Sir, good afternoon, sir.
Good afternoon. Good afternoon.
Sir, good afternoon, sir.
Okay,
Good afternoon. Good afternoon.
তাহলে শুরু করছি।
হ্যাঁ,
Okay. So, sir, good afternoon,
good afternoon, everyone.
Okay, thank you for
Good
joining us
afternoon. Good afternoon.
তাহলে শুরু করছি today's webinar organized by
হ্যাঁ।
Department of Computer Science, New Alipore College. So, my name is Sir. Good afternoon,
Good afternoon,
sir.
everyone.
Okay, I am an assistant. Thank you for
Good
joining the
afternoon.
Department of
Good
Computer
afternoon.
Science. তাহলে শুরু করছি
today's
and the
webinar
organizing member organized by
হ্যাঁ।
Department of
this
Computer
webinar
Science, New
named
Alipore College.
e-EVOLVERE
So,
2023.
my name is Sir. Good afternoon,
Good
sir.
afternoon, everyone.
We have
Okay,
started.
I am an assistant.
Thank you for joining the
Good
Department of
afternoon. Good
Computer
afternoon.
Science. তাহলে
in
শুরু
the
করছি
year of
and the
webinar
organizing member
2018,
organized by
and
Department
this
of Computer
webinar
Science.
is extremely happy
named
to get the
e-EVOLVERE
opportunity to organize
2023. My name is
for
Sir.
the
Good afternoon,
fifth
sir.
time.
We have
Now,
started
I am an assistant
on the
for
topic
Good
of the
afternoon.
of
Good
winners.
afternoon.
তাহলে
in
শুরু
the
করছি
year of
and the
webinar
organizing
2018,
member organized by
and
Department
this
of Computer
firstly, I
Science.
would like to
named
welcome
e-EVOLVERE
our opportunity to organize
2023. My
Dr.
name is
Joyce
Sir.
sir.
Good afternoon,
Good
sir.
afternoon,
We
everyone.
have
It has always
started
been a continuous
on the
process
topic
for
of
us
the
to
afternoon.
reach to
Good
our
afternoon.
winners. In the year
and
of
the
webinar, I would like to
organizing member
welcome
organized by
and
Department of Computer
firstly, I would
Science.
like to
named
welcome our opportunity to
e-EVOLVERE
organize
2023. My name is
Dr.
Sir. Good
Joyce
afternoon,
sir.
sir. We have
It has always
started
been a continuous
on the
process
topic
for
of
us
the
to reach to
Good
our
afternoon.
winners. In the
and
year
the
of webinar, I would like to welcome
organized by
and
Department of Computer
firstly, I
Science.
would like to
named
welcome our
e-EVOLVERE
opportunity to organize
2023.
for
My name is
Dr. Joyce
Sir. Good afternoon, sir.
It
We have
has always
started
been a continuous
on the
process
topic
for
of
us
the
to reach to our
Good
winners.
afternoon.
In the year of webinar, I would like to welcome
organized by
and
Department of Computer
firstly, I would like to welcome our opportunity to organize for
I would like to
Dr. Joyce
Sir. Good afternoon, sir.
It
We have
has always
started
been a continuous
on the
process
topic
for
of
us
the
to reach to our winners. In the year of webinar, I would like to welcome
organized by Department of
firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
in the year of webinar, I would like to welcome
organized by Department of
firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
in the year of webinar, I would like to welcome
organized by Department of
firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
in the year of webinar, I would like to welcome
organized by Department of
firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
in the year of webinar, I would like to welcome
organized by Department of
firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
I would like to welcome firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
I would like to welcome firstly, I would like to welcome our opportunity to organize for
I would like to Sir. Good afternoon, sir. We have started on the topic of the
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We have started on the topic of
I would like to welcome firstly, I would like to welcome our opportunity
Now I would like to Sir. Good afternoon, sir. We Now I would like to sir, good afternoon, sir. I'm audible. Now I would like to sir, good afternoon, sir. I'm audible. Now I would like to sir, good afternoon, sir. I'm audible. Now I would like to sir, good afternoon, sir. I'm audible. Now I would like to sir, good afternoon, sir. I'm audible. I would like to take now I would like to sir, good afternoon, sir. I'm audible. I would like to take now I would like to sir, good afternoon, sir. I'm audible. I would like to take now I would like to sir, good afternoon, sir. I'm audible. I would like to take now I would like to sir, good afternoon, sir. I'm audible. I would like to take now I would like to sir, I'm audible. I would like to take
Is it visible? Everything is okay?
ভিজুয়াল দেখা যাচ্ছে।
Okay, so it's actually a great pleasure for me to here today to discuss the fascinating world of computational landscape in 3D genomics. Okay, so with a special focus on some network analysis. So it's actually a great pleasure for me to here today to discuss the fascinating world of computational landscape in 3D genomics. Okay, so with a special focus on some network analysis. So it's actually a great pleasure for me to before starting to discuss the fascinating applied of computational landscape in 3D genomics. Okay, so with a special focus on problems some network analysis. So it's actually before starting, what type of data we are working with, like what are the problems that we are trying to. Okay, so especially focus problems in education. So it's actually before as in what type of data field of genomics, like what are the advanced particularly with the integration with especially focus problems in education. So it's actually before as in what type of field of genomics, like what are the advanced particularly with the integration especially those problems in higher dimensions. So it's actually and before as in what type of field of genomics, like what are the advanced particularly with the those problems in higher dimensions. So it's actually and before as in what type of field of genomics, like what are the advanced particularly with the complete those problems in higher dimensions. So it's actually and before as in what type of field of genomics, like what are the advanced particularly with the complete those problems in higher dimensions. So it's actually and as in what type of field of genomics, like what are the advanced particularly with the complete those problems in higher dimensions. So it's actually the model typically as in what type of field of genomics, like what are the advanced particularly with the complete those problems in higher dimensions. So it's actually the model typically as in what type of field of genomics, like what are the advanced particularly with the the model typically as in what type of field of genomics, like what are the advanced particularly with the model typically as in what type of field of genomics, like what are the of DNA can be thought of as string of composed of 4 letters. It could have been any possibility, as you can see an example of there. So as it consists of DNA can be thought of as string double helix composed of 4 letters. So each strand has a DNA any possibility as a direction, as you can see 3 prime to 5 prime there, and so to both ends was representing this one. So as it consists of DNA can be thought of as string of brief about the DNA, because strand has a DNA as a direction, as you can see 3 prime to 5 prime information while we are working with this to both ends was data in terms of so as it consists of DNA can be thought of as computational brief about the DNA. Each strand has a DNA as a direction, as you can see 3 prime to 5 prime information while later we are so both ends was data in terms of so as it consists of DNA next computational brief about the DNA. Each strand has a DNA provide a brief where the DNA is information as later we are so both ends data in terms of so as it consists of next we are studying those brief about the stock and we are going to provide a brief where the DNA is information as later we are going to so both ends data in terms of so as it consists of next we are studying those brief about the stock and we are going to provide a brief where the DNA is information as later we are going to so both ends data in terms of so as it comes to next we are studying those brief about the stock and we are going to provide a brief where the DNA is so both ends data in terms of so as it comes to next we are studying those brief about the stock and we are going to provide a brief where the DNA is before starting the further data in terms of so as it comes to next we are studying those brief about the stock and we are going to provide a brief where the DNA is before starting the further data in terms of so as it comes to next we are studying those brief about the stock and we are going to provide a brief where the DNA is before starting the further data in terms of so as it comes to next we are studying those brief about the stock and we are going to provide a brief where the DNA is before starting next one is then a chromosome consists of a nucleosome plus a histone. Then it constructs the next level where this then a chromosome consists of a nucleosome plus a histone and make a coil-like structure. Then it constructs chromatosomes. The next level, the chromatosomes, which folds up to the produce of 30 nanometers of fibers. Then a chromosome consists of a nucleosome 6 level plus a and make a coil, the loop averaging. Then it constructs nanometers in the next level, the chromatosomes, where is working up to the produce of 30 nanometers of fibers. Then a chromosome consists of a nucleosome 6 level plus a and make a coil, the loop averaging. Then it constructs nanometers in the next level, the chromosomes, where is working up to the mapping fibers. Then a chromosome working with computational landscape. Information, they are constructing the loops. So in linearly you can say this is the coil, one is coiled, and the same one is considering by some other factors I'll discussing later. Information, they are constructing the loops. Other factors so there that in linearly you can say to come together to is one is genomic regions. And so basic level, you can consider this as a loop. By some other factors, I'll discuss later. This 300 nanometer fibers are compressed and folded into 450 nanometer wide fiber. One is genomic regions. The seventh basic level, you can consider this as a loop. So some other factors, I'll discuss later. 300 nanometer fibers are compressed and folded into finally 450 nanometer wide fiber. One is genomic regions. The seventh basic level, you can consider this as a loop. So some other factors, I'll discuss later. 300 nanometer fibers are compressed and folded in finite to the side of white fiber. So this is the basic structure. So basic level is a loop. So some other factors. 300 nanometer fibers are compressed and folded in finite to the side of white fiber. So this is the basic structure. So basic level is a loop. So some other factors. 300 nanometer fibers are compressed and folded in finite to the side of white fiber. So this is the basic structure. That basic level how loop. So some other factors. In other nanometer fibers are compressed and folded finite to the side of white fiber. So this is the basic that there is the basic how loop. So some other factors. In other nanometer fibers are compressed and folded finite to the side of white fiber. So this is the basic that there is the basic how loop. Some other factors. In other nanometer fibers are compressed and folded finite to the side of white fiber. So this is the basic that there is the basic how loop. Some other factors. In other nanometer fibers are compressed and folded finite to the side of white fiber. So this is the basic that there is the basic how loop. Some other factors. In other nanometer fibers are compressed and folded finite to the side of white fiber. So this is initially there is the basic how other in other nanometer fibers are compressed and folded finite to the side of white fiber. So this is initially there is the basic how other in other nanometer fibers are compressed and folded finite to the side of white fiber. So this is initially there is the basic how fibers. Then obviously the nucleosome fibers was constructed with the histone modification. These are the histone modification IDs that will construct this looping. then the compaction of DNA binding proteins with the histone modification. This is the most important one. IDs chromosome level that will several architectural proteins that will construct this protein. Then the compaction of DNA binding proteins is made by modification. This is the most important. That second level with the IDs. When a chromosome level that will several architectural proteins that will construct this, then the compaction of DNA binding proteins is made by modification. This is the most important. That second level with the IDs. When a chromosome level that will several architectural
and some other factors,
then the compaction of DNA binding proteins is made by modification. This is the most important. Second level with the IDs. When a chromosome level that will several architectural
and some other factors,
then the compaction of DNA binding proteins is made by modification. This is the most important. Second level with the IDs. When a chromosome level that will several
and some other factors,
then the compaction several that will CCV extract second level with the IDs. When a chromosome level that will several
and some other factors,
then the compaction several that will CCV extract second level with the IDs. When a chromosome level that will several
and some other factors,
then the compaction several that will CCV extract second level with the IDs.
several and some other factors,
Then the compaction several that will CCV
several and some other factors, and some other factors, and some other
CTCF protein. So due to that CTCF protein, there is interaction also there. So this very basic level information we can get it. So okay, there is a comparative discussion of Chapet and Hi-C, where Chapet is obviously always find the chromatin immunoprecipitation due to the CTCF protein or other protein factors. Maybe it can be RNA poly to RNA polymerase. And Hi-C is chromosome scale interactions. So now, how to analyze those 3D organization in terms of network? Because as I told that in previously, I'll describe some of the network-based approaches to assess those 3D organization, and second phase will show some of the deep learning approaches that will find and try to find the interactions level prediction from the very basic information as a DNA sequence without acquiring any other 3D information, but from basic sequence DNA sequences. So these two approaches we have discussed here. So initially, for assess those 3D organizations, so due to the sequencing, we have all the informations where the gene located, what are the regulatory elements, where the domain boundary within the domain, how it was interact, and how the CCDs or TADs are there. So there is a pictorial representation of there within the chromosome. We can find the chromosomal territories. Within the chromosomal territories, the TADs are there. And there is a factor, a domain boundary factor here, there. So with that information, where we can locate the enhancer, cohesion, CTCF, the enhancer are obviously the promoter that activates the gene. So all that information that we have now, how can we analyze this architecture in terms of network? So as we know that network is an intuitive way to pictorially represent elements and their interactions in many complex systems. There is several applications in now in social science, earlier in proteomic interactions. Similarly, we can assess the network of genomic in terms of network representation. So for that, whenever again we need to think about the linear organization, linear 3D organization in terms of linear mode. So we have a total genomic sequence. From that, the sequence read, we have a particular region from that sequence. So several regions will be there. Within the region, there is a information of interactions. So simply we can represent in terms of network, where these genomic regions will be considered as a node, and the interactions will be considered as a S. Obviously, there is a weight of the S because the reads are at population level. So maximum reads provides maximum possibility of interaction on that region. So introduce some network or topological properties to assess those network, how it was there. But the most important thing is how we can represent this network in more informative way, where all the factors will be involved. Because whenever we need to acquire a complete information regarding a complete network system, we need to incorporate all that factors that mediated that interactions, that mediated that the organization of the 3D in spatial mode. So we need to include those. So how can we do this one? So that was also a challenging things. So at layer by layer, we have incorporated the information to assess those networks. So here is an example of a sub-region from the chromosome, chromatin. So if we start, so the network you can think as a start and stop in terms of linear mode. So the locations of a genome, if we start from 5 kb resolution to 10 kb resolution, 5,000 base pair position to the 10,000 base pair position. So we can start as a start, consider this as start point and the end point for sake of linearity consideration. So here, so otherwise there is no start and stop in terms of 3D organization. So if we can start this this region and consider all the interactions up to this region, and then this is a linear connections. That means if we can trace the genome, it will be a linear mode. Then these connections, it will be looks like this one. but in 3D there's some other factors. So while representing the network, we can simply see that this is not linear in terms of their position wise connectivity, but they have cross connections. So how we construct this one? So initially, for that consideration, we start with at a domain level. Domain means the chromosome conformation captured domain, or we can see the TAD domain, topologically associated domain within the 3D organizations. So at that scale, we have started. So within a, if we consider as a single CCD, within a CCD, how the networks works. So we generate all the networks from all particular possible CCDs. This work is started with the CTCF chapter data, where the interactions was mediated by CTCF protein. Then we construct multi-omic networks, including both genomic and proteomic one, because most of the cases, proteomic network was assessed in terms of functionality, in terms of network science, to extract the functionality, and it was shows a significant improvement on that region. So that motivates us to introduce the initial level, the proteomic interactions. And first of all, we assess that the CCD specific subnetworks, what are the properties of them, whether they are random network or not. Is there any significance of their randomness, or they have some other significance on that type of structure? That was assessed in terms of concentration of SNPs, single nucleotide proteins, that causes some disease. SNPs are, as a result, I'll show that at the end. With this network architecture, we found some concentrated region of SNPs within the TAD regions by their topological properties, and they are associated with some autoimmune diseases. So there is a pipeline of that. So I can describe this like a, so for that we have worked with the cell lines. So GM12878 is the lymphoblastic cell line name. So we extract all the interactions, and before one more thing, before starting this network-based approach, each regions here we named as anchors. So each region that was interact with the other one, that was named as anchors. Anchors also identified in terms of sub regions that occurs in all populations. So there is a specific algorithms was there that identify those anchors in terms of their histone modification regions, the peak values, the regions that continuously interact with the other regions. So for simplicity, you can consider a specific region as anchor, and we represent those anchors as a node and the interactions as the edge. So by their coordinate, we can find this. So all total, the networks initially was found with 80K interactions for lymphoblastoid cell lines, and composed of almost 2K CCD regions, the domains, and total interactions across all domains are 80K. So while these interactions was converted, we have an anchor network. Then we try to construct the genomic network, because the anchors are specific regions that does not signify that whether there is a gene or not. So we try to map these in terms of coding genes, because that genomic region, which is correlated with the coding regions, so linearly we try to interact which one is closest to this anchor that will be assigned to there, because sometimes it could be within the anchor region, that could be outside the anchor region. But why we are connecting this one? Because there are some protein factors or some other factors that will mediate those or encode those coding regions. So there is influence on that genomic regions that will encode this, or that codon region will be converted into protein. So there is influence on that. That's why it was linked to there. So that by simply a threshold based Euclidean distance, we assign those gene in terms of anchors. So we'll achieve at the next level the genomic interactions. Then we add, because as we have a link that this particular gene has coded into a particular protein, so the link is straightforward. So we try to construct those links from gene to protein. Then the protein networks is available experimentally. Protein interactions are available, so we can connect those interactions, verified interactions, and then we construct the meta network. So, and finally we have also include other factors, but with this network we try to assess the properties of that network, how they behave. So there is a example of network preparation. This is whole genome network. It's a simple one. This is a node, the interactions. So it's a simple pictorial view for the GM12878 cell lines. Then we segregate those interactions, because we are not a interchromosomal region. We are working with this chromosomal region because we need to go within the CCD up to that region. So within the CCD, we try to find the subgraphs. These interactions are purely not the linear one, as per the 3D genome wise, the interactions. Then we try to find those anchor networks and convert into a closest genomic network. And this is total data flow for that. How we converted anchor network to genomic network, and finally the proteomic network. So one thing that we found, this is up to the genomic network, then there is a proteomic network. By nature, these genomic interacting networks are less dense compared to the PPI networks. This is obvious actually, because one encoding gene could have a multiple protein due to the variants. So, and the proteins are more effective in terms of functionality, and so due to that particular functions they interact each others, and basically these are more dense in by the properties. So degree distribution plots have actually shows that the proteomic is a denser one. So next, this is the overall network for that. So nucleus, then chromatin network, gene association network. At that level, this is anchor, gene, and protein. Now one by one we add the other factors in this network later. So whenever we have this network, we try to construct a vertex induced mode of representation, where and try to find any two nodes within a CCD, if they are connected in genomic network, whether they are connected or not. But we found some fine observation that any two nodes from the genomic side, if it belongs to a particular chromatin captured domain, and we try to find the coding proteins in terms of PPI, they are mostly connected by elongating the number of nodes, intermediate nodes, up to the 4 node. So there is a plot for all subnetworks in a single mode. If one genomic pair of interactions is converted in terms of PPI, protein-protein interactions, it's not likely to interact directly in terms of PPI, but there are some intermediate nodes. So at 0 level, the less number of subnetworks are connected. At 1 level is increased, and 2 level almost all the networks are connected, some of them. And while we along this up to the 4 intermediate nodes, all the more interesting thing is all the genomic interactions are also connected, and it's a complete mode in terms of PPI. Similarly, we assess those subnetworks in terms of some topological properties, closeness centrality, betweenness centrality, and the clustering coefficients, how they behaves in that representation. And next, we exemplify whether the property of this network are random or not. For that purpose, we have generated two different versions of random networks, where putting the constraints on the number of edges, and the second one where the number of constraints was put on both node and edges. So with 100 iterations of the randomness, we compare them with the exact networks, and significantly they are not random. While we comparing this, they significantly differ from each other in all three properties. So this is a complete representation of complete genome in terms of closeness centrality, betweenness centrality, and the distribution clustering coefficients. In all situations, both the random networks are significantly differ from the exact network. So we can say that these architectures are special, the organizations are special, not a random one. So this is from the computational side or statistical side, we can identify this property. But is there any speciality of that non-randomness? That we need to find. So for that purpose, we have tested some of the regions while mapping with the geo and pathways, gene ontological term, basically the annotations of the function. And obviously the pathways. For this pathway, we have used cake, reactome, and uni pathway. And the most important thing is they're associated with, we have tested this with the autoimmune diseases, whether what number of CCDs or the subgraphs are significantly enriched with these SNPs that are related with the autoimmune diseases. So more than 80% autoimmune diseases was associated with SNPs that located within the network region. There is less number of SNPs that are associated with the autoimmune disease that are located in the interchromosomal regions, but it's very low with the significant high number of p-values. And also introduce the structural variants. I'm not getting in details of this as we. So they are also significant for the disease variability. So that's why we have incorporated the structural variants. And also later we have introduced the directionality into the PPI. So how the information flows or disease flows from community network community one community to another one. So that has a significant role of directionality of PPI. That was also incorporated in the meta network representation. So this is the overall architecture of the network assessment, where we started the anchor, then introduce gene, protein, the network. They are not random in nature. Then introduce gene ontology, directionality, and PPI, the other pathways, the structural variants, to find whether they are significant. These networks are significant or not. The organizations of the 3D networks are significant or not. So finally, we show that for a particular regions, which shows a significant number of enrichment in terms of disease, which are within a non-random interacting network that was extracted from the 3D genome. So later we have also found the functionality. So till now we have tested on a single cell lines, GM lymphoblastoid cell lines, GM12878, and in more advancement mode we have tried to find out whether the functionality is conserved within the different cell or not, or similar cell types or not. considering those meta networks. So here is a heat map plot. We have tested with 24 blood cell lines. I think these are not visible. There is a tissue type for different 24 blood cell types, but we found that at different level the functionality is conserved, and it was compact where we adding those layers into the network. So that's all about the network assessment. Some other additional task was going on. So now if we can go to the classification problem, where so first I try to figure out the problem in terms of classification, then we go to the particular problem that we have resolved till now. So we have DNA sequence, we have the structural informations, and we have some gene expressions, that means phenotype informations. And at different tissue level the interaction information we have, but we need a mapping from genotype to phenotype. So initially one step is there where we considering the 3D interactions, and then we find the path from genotype to phenotype, whether that interactions at different cellular tissue level can find the expression level for a particular gene or not. That is one point. And in that situation we need that interactions at different cell tissue type. But the one more step before that, if we have the DNA sequence, are we able to find those tissue level interactions from that sequence? Obviously that tissue have some protein factors or may have some different protein factors. So considering those protein factors, the modification histone modification factors, and obviously locating those enhancer promoter locations, initially in that case considering the promoter locations, whether we are able to find that interactions or chromatin interactions at different tissue level or not, with variable varying these tissue types or the cell types. So that is the problem. So for that, initially we try to find the interactions at the population level for a particular cell types. Obviously in first case we have worked with, as we have already working with the lymphoblastic cell line. First situation we have tried to find this in terms of lymphoblastic cell line, then we try to find it cross cell line or cross tissue level. So there are some biological questions in order to find those interactions. Before that interactions, whether it was possible to find the proximal or core promoter regions. Now in that situation we need to consider we have only one information of DNA sequence, whether from that DNA sequence it is possible to find that particular protein factors for proximal regions, transcription factor that will helps for binding sites, and their functioning genetic variations. So initially we need some learning strategy for that. So for that case we need to provide some annotations. So all the informations like enhancer, RNA pol to core promoter, where the gene body, that need to be localized in the genomic regions in linear mode, because we are starting with that sequence. Then we need to explore all one by one at the layer by layer in silico level. So we need to verify whether it is fine or not. For that the annotations is required. So all times it is not possible to find the annotations. If it is possible and easily accessible, then then there is no requirements of the in silico models. So for that case, the bioinformatics scientists have make bias towards the NLP based approaches, where they can use the DNA sequence as a language model or not. From that sequence, if we can generate the language, and we can see the advancement of the NLPs in terms of this language modeling and the GPT versions, as we know. So based on that concepts, try to construct the language model from the genome, human genome sequencing, the reference sequence model. So for that, it was more effective in recent days, the bidirectional encoder representations based transformer model that will construct the DNA language in genome. that will try to identify the non-coding DNA into one fundamental proposition in terms of their particular level of classification, whether this is a proximal region, this is a binding factor, there is a binding factor, there is a promoter, whether there is a that multitasking problem can be resolved if we have that language label, language model from the DNA sequence. So three basic expectation from the language model is that whether it can take all the contextual information into account to distinguish polymerase cis regulatory elements, that will consider all those things, developed a generic understanding that can be transferred into various tasks once we have this language model, and then we can use those language model in terms of various fine-tuning tasks. And the most important thing is that will generalize the reference sequence as for all at high resolution. We don't have any the sufficient label data is not available at that situation. So in that case, this is more suitable to generate to get a language model from the DNA. So for that, several approaches was there. CNN based model and RNN based model was there, but all of them requires labeled information. And for that case, the people are more motivated to construct the language model with the BART architecture. So obviously the motivation comes from the the paper of Vasan in 2017, attention all you need. You can check the paper. That's widely now a huge impact of that paper in NLP now, obviously in terms of bioinformatics as well. So I'm not getting in details of that, but how we can represent the DNA sequence in terms of this language model, I try to explain in step by step in more simple way. So the first level is that as we know that in NLP, while they constructing the language model, they consider each and every word as a and all those the language model have much more number of vocabularies where each word will be stored. So as the powerful of number of words as the powerful of the language model. So in terms of DNA sequence, we have a we have a most interesting thing in that case, that we can generate our words in a fixed within a range. We know the range of that sequence and we can maximize this. So for that case, first of all, how we can generate we can extract like a word information. For that case, from the very basic original sequence, we try to extract some word in different representation of term or from tokenization. We can say that tokenization, which is nothing but a k-mer, overlapping k-mer from a sequence. What is overlapping k-mer? So at the payload level, you can find this. There is a 3-letter k-mers. So let us assume this is a word as it was words in terms of NLP. So one word will be tokenized. Like here, the fixed length word will be there. And as we know, we have 4 letters. So 4 cube number of possible words can have been, that will be the vocabulary size. Similarly, if we extend those length of the k-mers, accordingly it will be larger, the vocabulary size. But it was tested that with the 6 length of k-mer, it was performing better. Here is an example of 3 letters k-mers. So at the very beginning level, the original sequence was discretized in terms of subsequences of k-length k-mers. Then there is a pre-training stage that will generate the language model, keeping some of them skipping from the normal generated sequence as a masking one. Then it try to find the token embedding. Then it requires a positional embedding, because the position informations for the sequence is very important for localization of the genomic regions. So this is more suitable for the positional embedding as it was required in terms of grammar representation in NLPs. So that step, one by one, the input embedding was constructed for that language model. I mean, if you consider this language model at the first beginning level, where the pre-training, so we can devise this task where first one is language model, that is nothing but a pre-training one, and the second one will be the task specific classification. So second, there is a comparative discussion in terms of CNN and RNN and in terms of transformer. We're not going to details about this. Once we have this input embedding, then there is a bidirectional transformer architecture was there. Then it construct the network, and finally it introduce some self-attention to identify most informative k-mers within that input sequences. That helps in terms of later fine-tuning steps, where we have to find the problem of motif regions within a DNA sequence that highest intention self-attention k-mers have a significant number of enrichment with this motif. So that also a second level efficiency of those networks. So here use multi-head attention and softmax function for the head computation, final level head computations. So I'm not getting details of that discussions. So in a brief way, we can find earlier, if you can think about this, the traditional model was that there is a starting point. The model will be learning with the classification, where we have the all the label data. Then it will be trained and it will be classified. But in this step, it will generate some first language model, where the task is only identify those masking regions at the sequence level, with that it can perfectly identify those, receive the attention information from those networks. That will help in terms of fine-tuning. And once we have this model, now we can use those language model in terms of several tasks. That is fine, task specific fine-tuning. So there could have been multiple models that will incorporate that informations from the language model, and that tuning requires a classification task, particular task. So accordingly it will classify. So with this language model, this is the very basic structure of the transformer model. So there are consecutive 12 transformer layers was there. So all of them, this is the basic structure where the classification is done in two way. One is mask token classification, so that is similar to the language model task. And second one is sequence level classification. Token level classification means what identification, and the sequence is total combinations of the words in terms of sentence here. But in terms of NLPs, but in terms of genomic sequence, the motif identification like problems was there. So finally, with this language model, we try to construct at the first level, we constructed a deep hybrid learning strategy, because with this pre-training model, we generate some of the fine-tuning to find those interactions, the chromatin loops, as we have discussed earlier. So we have sequence. This is a one-step jump from sequence to chromatin loop, very basic architecture of those. And whenever we have generate all those loops within a particular region, then there will be a polymer modeling that will incorporate this in silico chromatin loops to generate the architectures. And similar at the second level, it will find the compartments and the CCTs. So that is the steps we have devised. So at several level we are working on there. So initially, this is once again, this is a genomic sequence. The regions was identified, and the sequence was there. All the sequence was connected which have a loop. So if there is a loop, then the two sequence, this sequence length and the sequence length will be there. So this is basic input representation for the fine tuning task. So initially with the reference sequence, the model was generated, pre-trained model. With this model, then the fine tuning task is here only to find the interactions where two specific given sequence was there. Then the model will identify whether they are interact or not. No other factors was inputted in this network. So very basically we have considered the sequence. So initially we construct without any other the fine-tuning label, only the prediction scores, and additionally we generate some of the machine learning based models considering different k-merge sequences from this sequence, and the prediction was made, and finally it was ensembled with the machine learning based approach and the DNA-part fine-tuning approach to find the deep hybrid learning. So almost it provides us state-of-the-art approaches up to the significantly better in compared to the state-of-the-art approaches. So recently it was published in Quants Biology. It has a simple performance where we have exemplified all KML length specific accuracy on all ML-based approaches. For ML-based approaches, we have used SPM, RF, and KNN. And obviously the DNA-part pre-training was there. So here is the performance of individual level and all the machine learning and the deep learning based approach. However, the DNA-part has a higher accuracy compared to the all machine learning. And finally, the DHL model have surpassed all the performances while comparing the DNA-part and all ML-based approaches. Here, this interactions was tested with two protein factors, CTCF and RNA-pol2, RNA polymerase data. The interaction that was mediated by the CTCF and RNA-pol2, and the experiment was done with the lymphoblastoid cell line. So at the next level, we try to exemplify whether the performance is consistent over different cell types or not, or different experimental data or not, different protein factors or not. So including considering all those variability, we have modeled the next level for the predictions, where some modifications or advancement was there that also helps to identify the loops at higher accuracy. So for that case, here is a pipeline was there, total workflow. So we have introduced different bioinformatics pipeline that will generate those data sets, interaction data sets, across different cellular tissue types of lymphoblastic cell. So that provides us the variability of the data. So similarly, here also consider the interactions. Obviously, for the prediction, the interactions are based on their anchors. So while we are considering the anchors, but while considering these anchors as input, that is nothing but a part of DNA sequence. So we have a DNA sequence information only. Then we need to find this one. So considering all those bioinformatics pipeline, there are several variations of data sets will be there. Now we do some modifications in terms of training, before the training, whether there is a noise or not, because the learning method will be biased if we have a noise in data or not. So for that case, if we have let us assume we are considering the CTCF protein factors. That means the interactions that was mediated by these protein factors. Okay, so due to this error in the reading technology, there could have been some regions that was identified as interactions, but there is no protein factors of that particular protein types. So how we can assess those there is interactions or not? So for that we have done some of the filtering, whether it has a peak or not on a particular anchor region. So initially we have a DNA sequence of all complete genome. Then we try to find the peaks. We have all the anchors. We find whether there is a peak or not. Peak is a signal for a particular protein factors. Whether the enrichment is nothing but whether the peaks was located within the anchor region or not. That is. Next, we try to find the CTCF motif. We have developed an algorithm that will find the CTCF motif with the PMF factors that can generate the motif regions from only the information of sequence. Now, one of the problem in current BERT model is that it cannot access because it requires a huge memory space in computations. So till now, it can consider only 512 positional encoding. So we need to make it limited the input sequences. So initially, the anchor regions is varied from 50 kb to 100 kb. 1000, yes, 100 kb. So and while we are considering this BERT model, the tokenization was done up to the 512 number of positional encoding, so that number of k-mers can be generated. So for that case, we need to reduce the input.
অনুপ, শোনা যাচ্ছে না, মনে হয় মিউট আছে। কিছু নেটওয়ার্কের প্রবলেম হয়েছে বোধহয়, কারণ তোমার স্লাইডও দেখা যাচ্ছে না।
মিউটেড রয়েছে।
হ্যাঁ, মানে স্লাইডটাও দেখা যাচ্ছে না। ঠিক আছে এবার।
So I'm not sure where it was muted. So for that dimension reductions, we try to find the motif regions within the anchor of 250 bp, both on the both anchors, those who were interacting, and then generate the k-mers from the motif, and the transfer model was there. And also it was incorporate the language model from the pre-trained language model of the similar k-mer size. Then it was validated with self and cross cell testing. The validation was done not in terms of statistical validations, the statistical metrics. We also introduced the polymer model for the verify whether the polymer model in 3D are similar, more or less similar in terms of their generated model generated from the in silico mode and the real data. So two level of validations was done and for the advancement of the in silico pipeline. So here is the details of the, I'm not going to details of that. This is a statics of different cell types and different protein factors. One more thing, the most important thing here, we have considered two types of protein factors. One is CT-CIP and cohesion. So this is lymphoblastocyte, all of them are lymphoblastocyte cell lines, and these two is Parturian family, two different cell types. So they have initial anchors and interactions. From there, we try to find the initial label from the sequence. We find the motifs within the anchors. If both regions have the motifs, then we consider those data as a positive interactions. And similarly, we generate the negative interactions. This is most important one. So the negative data interactions is most impactful in terms of this type of classification while considering the pair input data. So there are three types of negative class construction, or we can say the test classes: complex 1, complexity 2, and complexity 3. In general, the random negative data was generated. If you consider this positive data as a graph, that means two nodes, because this is a pair input method. Two pair of input data construct a single data, and the class label is only for those pair of data. Okay, so the problem is that whenever we consider the negative data, we consider all the positive data and generate all possible pair from the individual data sets and remove those positive data. The remaining data will be the negative one. That means nothing but the, if we construct the the negative data, that is the basic consideration earlier in terms of PPI as well as any interactions data that was generated, because there is no data sets for the negative data in terms of pair input data. So there are some couple of data sets in PPI, but I have not any idea about this genomic. I could not find it any negative data in terms of, because always it was found that in an experiment, whether two regions are interact or not, or whether two spatial regions are closer or not, whether two regions always remains distant or not, that was not identified. So we need to find some strategy to extract those negative data. So this is an important thing in terms of learning. So all the experimental methods till now, except some of them, have employed these random negative generations. But there are some problems while generating this random data, negative data, because in chromatin interactions, there is some task in recent bioinformatics 22. They have shown that there is some transitive relationship while three particular regions, they have interactions, both have different interactions. If we realize this in terms of 3D, that means actually they will be A, B, C. A will interact with B and B will interact with C. So in random generation, A and C, there is no link, so it will generate as a negative one. But more than 40% interactions are like transitive interactions. In recent papers, they have shown that transitivity also finds some significant number of interactions. So there is a problem if you consider this type of interactions as a negative one. So classifier got biased. So there are some several filtrations for that. There is no interactions will be there in up to level 4 for the negative data generations. And obviously one more thing, if spatially two regions are distant, then there is a less probability of interacting together. So while we considering the negative data, first of them, they are not linked with the level up to level 4 in the connected network, and they are distantly located. So in that way, the negative data was generated for that. So this is overall pipeline for that model. So where there is no individual model, so we have different variation of data with different protein factors. So the data will be generated in various mode. So every combinations there will be a model for that. Similar embedding was there because we have used here also the pre-trained model of pre-DNA bird. So, and 12 transformer block was there, considering the multi-head attentions, and finally it generates the predictive model. Now, so we have two different variation of cell types, lipoblastoid, and two different variations of protein factors. Is it visible?
দেখা যাচ্ছে।
Okay. So here the row wise represent the training factors and this column wise it represent the test factors. So across all the cell types, while considering the, because it should, the normal concept is that if we have if we consider the protein factors, I mean similar protein factors enforce always a similar type of predictions, because the nature of the protein factors is more or less similar. And similarly, while considering the CTCF, so it should be, the performance should be higher where the different variation of other cells of similar protein factors. But we found that this is consistent over all the data. So all the cell lines, all the protein factors as well. And also the performance has been enhanced to our previous model as well, which was the best state of compared to the state of the art initially. But here it was improved because of two important things that we have taken. One is the motif regions, because in earlier cases we are considering the midpoint base region within the anchors, but there could have been the, obviously the possibility was there that we have missed the main informative factors from the sequence. So for that case we have introduced here a motif finding before going to the BART architecture, that was find the more informative region within the, that localize the CTCF motif regions. And for the question we found the peak regions. So centering the peak and CTCF motif, we extend both side 125 125 altogether 250, and both anchors will be like 250 250. So in that way it helps us to improve the performance as well. So this is one type of statistical evaluation for our case. Now biologists will not be always pleased with this one. The statistical numbers are always are not all times favorable for them. So for that we have done some another validations. There is a this is a plot by ICB. It's easily Java application, standalone desktop application. You can check all of them if you have a particular type of input data. So we try to visualize those prediction scores. Here the upper side is real CTCF for GM12878 cell types, and the lower is the predicted one. So we have received some of the additional ages, although statistically they are not significant because they are false positive, but they have some significant. I'm describing one by one. So first of all, with this in silico model, we generate some polymer model in 3D in a volume constant way. Volume constant way means that it will be located within that volumetric regions, because all the polymer models was volume constant way generated. Yeah. So we generate the in silico model polymer for that and compare this polymer model with the normally generated polymer models from the real experimental interactions data. So one more thing before going to this polymer model based validation, these additional interactions was there, that this is the interesting thing for our case, that we identify those additional interactions. In fact, they are not a false positive one, because while we are considering cross cell type interactions, those interactions also available in other different cell type specific or tissue type specific experiment, and they have a huge intensity. I mean, the population level count was higher. That was validated. That means on that cell type based experiment, because this is a population level data, so there could have been the experimental error, or it could have been missed by those interactions in that experimental, and that was presented in other cell type specific interactions. So that's why it was more significant. And we have constructed all those in silico based information by considering at each step for each and every model. We have different cell type specific models, different protein factor specific model, but for each particular model we try to construct all 23, I mean not in 23 because the XY chromosome has less number of interactions, that's why we skip those as a model. So all 23 you can say like a cross validation of 23 sets. So each and every chromosome specific data was kept as a validation set, and each step we generate all the validated data. I mean, whenever it predicts a particular chromosome, that will be more totally in silico. So we generated 22 different times each and in particular chromosomes as an in silico model, and finally construct those uh, polymer models and compare there. So although this is not a task to compare all the low resolution level comparison of the polymer, because we need to find it as a higher resolution at the compartment level, more depth will be there. So up to this we have just validated this in terms of two types of real and predicted in silico polymer models. Here is this, although this the rotation is not fine. So a real polymer model with the volume, they have the similar number of, although the shape is not seems like identical, but the heat map is more or less similar. And we found because this is a more dense one, because it additionally found some of the interactions. But why it is significant? Because all the loops or physical association spatially that was identified, because and it shows up to 0.955 Pearson correlations between these two models. And the compartment level, I have not any figure now for the compartment level. The compartment level, it's more or less 98%, no, 0.98 Pearson correlation shows. So that's all my presentation, and here is my lab. Still we have two postdocs and several PhD students was there, and some of them also going to defense now. So you can check our lab as well. Some of the selected references was there, and
Okay, it's a nice talk, I think. So thank you, Dr. Anup, for this wonderful session. So from the participants, so if you have any question, then you can ask. So if you have any question, then you can ask. Okay, so also you can drop your question in Dropbox, yeah, chat box.
someone asked for slides. Okay, I can share it.
So anyone? Okay, I think. So if you have any question, okay, Prem. I saw somewhere DNA can be used to
Yeah.
store data, but how?
Okay. I think the question is DNA can be used to store data, but now. Okay. Yes, there are some technology was working on that, but I'm not sure whether they are successfully done or not. There's some DNA editing task was going on at animal level, but I'm not fully updated on that. Okay, the second one is the why BART when we have GPT now. Okay, for fine tuning. Yes, obviously that was also going on. Uh, we are also closely working on BioGPT because this is normal language level. So especially for biological sequence, the working is going on. At the very beginning level, it was with the BART architecture, it was working on that, and we hope the GPT can be BioGPT will be helpful for that. In more
Okay,
specific, the biological questions.
or anyone, any question? Okay, so I think we need to wrap our session here. Thank you all, your kind cooperation and patience. Specifically on behalf of our college, I would like to give my thanks to our speaker, Dr. Anup Kumar Haldar. He has managed some time for us from his busy schedule. We are extremely thankful to Dr. Anup. Hope to see you soon. And obviously, we all will, I think, eagerly wait to have such wonderful session again. Thank you. Thank you, Dr. Anup.
Thank you for the invitations. I also enjoyed.
Okay.
So,
Now we come to the end of our program today. So I give my gratitude to our respected principal sir and HOD for throughout support. Also, I would like to give my thank to the whole team. Without them, this webinar will not be possible to organize. Last but not the least, I would like to give my thanks to the participant to make this webinar successful. Thank you all, and once again, and then our HOD is now connected. So if so, I'm requesting our HOD to say something.
Hello.
Yeah, hello.
Earphone. Am I
Yes.
audible now?
Yeah, audible, audible.
Oh, sorry. First time at the beginning there was network issue. I could not attend actually, but right now I think it's okay. So I first of all say thank our honorable speaker, Dr. Kumar Haldar, for this um, means full of information. The presentation, his research insights, his domain knowledge and research findings for sharing those with us. So we thank our speaker. And next, I'd like to thank the organizing and the organizers. We are working as a team here, so everyone, every faculty member is partners involved. Yeah, so I'd like to thank all of you for organizing such a team. And I will especially thank the participants from different institutes, not our institute only, from different colleges, mostly in Kolkata. Um, the faculty members present here from different colleges, I'd like to all of you to, as you have joined this event and has made the success. So, I think you have been benefit or others from your interest from our this from this event, from the uh, speech of Dr. Halda. And, also, thank to my graduate students that you are here and have heard the session. I think I believe that you must have been benefited with all the information that Dr. Halda shared. So, in future, if you want to work in this field and want to do research work in this field, I'm sure that with help from Dr. Haldar also. Dr. Haldar, can I hear from you?
Yeah.
Yeah. How are you? I think you're fine. And after this long session, you are a little bit. Are you okay?
Yeah. Yes. Fine.
Oh, okay. Thank you. Thank you
It could have been
for.
better if it is.
Sorry.
It could have been better if it is in physical mode, I think.
But in that case, we won't get to this time particular this time as you are not here. But
Yeah,
actually,
that could have
from
been
the beginning, sorry.
that could have been an interactive session then, because
Yeah.
yeah.
Yes, yes. Actually, this time we are having some other assignments also. There are some preparations for uh, our activities also. From the beginning of eVolver, we used to organize it physically, and we had different events like competition, poster competitions, and that. So uh, we tried to make it physically, but this year it was not possible. So for next year, we'll definitely try to arrange it physically. And if you are here, then we'll try. I hope that you'll be with us. We'll be inviting you for another lecture. So if you are here, I hope you'll be here. So thank you so much.
Thank you.
So
Thank you.
that's it. Students, there are some questions from students. So I think all the questions have been attended. Isn't it, Saurabh?
Yeah,
Yes,
if you
yes.
have questions, you can mail me as well. I can, yeah.
Oh, okay. We have the mail ID of the speakers. Any queries and mail directly, and as he has, he will answer your queries. So thank you so much. Thank you all of you. Thank you. Special thanks to the speaker again. Thank you. And Saurabh also. Thank you, Saurabh, for
Okay.
conducting this session.
Okay. So we are closing this session. Thank you all. And Anup, thank you and bye.
The feedback is given in the chat box, I think.
Yes.
And it is
So
open for 10 minutes, I think. So intended, please give feedback, get the certificates.
okay. So all of you, please fill up the feedback forms already given in chat box. Okay. So after filling this feedback form, you will get the certificate. Okay. So feedback from link is already given in the chat box. Right. Okay. So I am closing the session.
Okay.